In this video tutorial, we are going to describe about the introductory part of the glycolytic pathway and the different phases of the pyruvate. So let's begin. What is glycolysis? Glycolysis is a 10-step enzyme catalyzed reaction where glucose is getting converted to pyruvate with the concomitant generation of two molecules of ATP. So why you are performing glycolysis in your body? Why are performing glycolysis in bacteria? Why glycolysis are performing in fungi, in all living animal or plant or whatever it is, all living body? Why are performing glycolysis? The actual answer is to generate ATP. Glycolysis produces the direct generation of two molecules of ATP and two molecules of NADH also. The NADH can be further utilized by a defined pathway, so that from the NADH you can also generate ATP. But if anyone asks you that what is the basic importance of glycolysis, the answer will be glycolysis is used where the glucose is getting converted to pyruvate with the concomitant generation of 2 ATP per glucose molecule. So it is a 10-step enzyme catalyzed reaction. So now the question is, from where glucose molecule is coming from? So the answer is glucose can be arised in your blood from the breakdown of higher polysaccharide, like you can use starch. Say starch is a plant polysaccharide. From the breakdown of starch or from the breakdown of glycogen can be stored in your body from glycogen. Say this is the glycogen molecule. So the uh, glycogen is normally stored in your body in the liver and in skeletal muscle. So from this polysaccharide, after hydrolysis of this polysaccharide by using specific enzyme, you can generate glucose molecule in your system. And glucose can be also synthesized from some non-carbohydrate precursor by a process called gluconeogenesis or neoglucogenesis. So gluconeogenesis. So just gluconeo. So gluconeogenesis is another procedure by which glucose can be generated in your system from non-carbohydrate source. So now the glucose is available in your blood, and you need to transport this glucose molecule into your cell. And for this, we have the cell membrane. Say this is the cell membrane. So this is the cell membrane, plasma membrane in our body, and this is the cell cytoplasm. So it is a cell cytoplasm. So it is the bloodstream. So glucose is getting into the cytoplasm by the help of specific transporter, such as GLUT receptor is present, GLUT, that is glucose transporter. GLU stands for glucose and T stands for transporter, and several sodium glucose pump is also present, that is Na plus glucose pump, Na plus glucose pump, just blue. That is Na plus glucose pump is also available by which glucose can get incorporated into your cytoplasm. And for glycolysis to proceed, glycolysis is a 10-step enzyme catalyzed reaction. All the enzymes of the glycolysis are present in the cell cytoplasm. So by this by the process of glycolysis, glucose is getting converted to pyruvate, and we are now going to the basic part of the glycolysis. So glycolysis starts with the glucose molecule, and you can see here the structure of the glucose. The glucose is present here, and glucose can be converted to pyruvate. Glucose can be converted to two molecules of pyruvate. Right, so this is the glucose, and glucose is getting converted to molecule of pyruvate, and an intermediate is produced here that is called fructose 1,6 bisphosphate, which is an important intermediate in the glycolysis. And by this process you are generating two molecule of ATP. So 2 ATP plus 2 pi is equal to 2 ATP. It is a net generation of 2 ATP, and we can also look that the 2 NAD plus molecule is used and the NADH molecule is getting produced. This NAD uses act as an oxidizing agent in glycolysis. For the continuation of glycolysis, we absolutely require this NAD plus. So by this process of glycolysis, we are generating 2 ATP, 2 NADH, and basically 2 molecule of pyruvate. So glucose is a 6 carbon sugar and pyruvate is a 3 carbon compound, and that is why after complete breakdown of glucose via the glycolysis, we are generating 2 molecule of pyruvate, because pyruvate is a 3 carbon compound. So now we are going to the different phases of pyruvate. There are definitely 3 different phases of pyruvates available, and 2 of them are anaerobic. The left one side, that is anaerobic homolactic fermentation. The absolute right one, that is anaerobic alcoholic fermentation. And there is another pathway that is called aerobic oxidation, which occurs exclusively in aerobic cell like us or like aerobic bacteria. So there are two anaerobic fates of pyruvate. One is called homolactic acid fermentation. Why it is called homolactic? Why not heterolactic? Because here only lactic acid is present. That is why it is called homolactic. No other compound, no other acidic compound like acetic acid or succinic acid or propionic acid, no other acidic compound is getting produced. That is the reason why it is called homolactic acid fermentation. It exclusively occurs in our skeletal muscle, which is an anaerobic tissue, and that is the reason why we are saying it is the anaerobic pathway. So this pathway is normally not occurring in all cell under a certain situation. When the rate of glycolysis increases significantly, then as you can see that for the glycolysis to continue, you require the NAD plus, and if you are increasing your muscular activity, in that case, all the NAD plus resides in your muscle will be converted to NADH. It means that no NAD plus will be remained in your system, right? So suppose you are increasing your muscular activity, you are increasing your skeletal muscle activity. In that case, the rate of glycolysis in your skeletal muscle will be increased than any other cell. And as a result, all the NAD plus that is present in your cell cytoplasm will be converted to NADH. That means no NAD plus is now present, or the concentration of NAD plus is extremely low to carry out the further reaction of glucose to pyruvate, that is glycolysis. So to immediate supply of NAD plus, what you need, the cell will convert this pyruvate to lactate, so that we can use this NADH for the conversion of pyruvate to lactate, and we are using NADH and we are converting NAD plus here. So it is just the reverse reaction what is happening in the glycolysis. In glycolysis, 2-NAD is getting converted to 2-NADH, but here 2-NADH is getting converted to 2-NAD plus. So it will occur only when the rate of glycolysis exceeds than the rate of other pathway, that is TCA cycle. Because we are aerobic, we require oxygen for our growth. So that means we have aerobic oxidation, we have citric acid cycle, we have oxidative phosphorylation, electron transportation, and all of these things are present in our body. So by this oxidative phosphorylation of citric acid cycle, we can convert NADH to NAD plus. But suppose in a situation when you are when the rate of glycolysis exceeds than the rate of TCA cycle of the oxidative phosphorylation or ETC, electron transport chain, in that case, immediately for the generation of NADH, this pyruvate is getting converted to lactate. Remember, lactate is a dead-end product in metabolism. It means that you cannot convert lactate to any other compound, any other metabolic pathway. So you have to go back from lactate to pyruvate, so that pyruvate can be incorporated further into either citric acid cycle or any other biochemical reaction. So lactate is a dead-end product in metabolism. It occurs only, that is the conversion of pyruvate to lactate occurs only when the rate of glycolysis exceeds than the rate of the TCA cycle or oxidative phosphorylation. So for the immediate generation of NADH, you are converting pyruvate to lactate. Lactate is a 3-carbon compound and pyruvate is also a 3-carbon compound. And it is an anaerobic process. Why? Because it is occurring in our skeletal muscle. There is another pathway, anaerobic pathway, which occurs exclusively in Saccharomyces cerevisiae, that is yeast, where the pyruvate is getting converted to ethanol and carbon dioxide. And it is known as the alcoholic fermentation. It is also anaerobic pathway. And yeast cell actually converts this pyruvate to ethanol and carbon dioxide. It is not a one-step process, it is actually a two-step process, where the first step is pyruvate is getting converted to acetaldehyde, and from acetaldehyde it is getting converted to ethanol. So by the first pathway, where pyruvate is getting converted to acetaldehyde, carbon dioxide is getting eliminated, and for this particular reaction you require an enzyme called pyruvate decarboxylase. So decarboxylase means you are removing carbon dioxide from a compound. So pyruvate is a 3-carbon compound and ethyl alcohol you are going to produce. Ethyl alcohol is a 2-carbon compound. So the first step is the removal of carbon dioxide catalyzed by an enzyme called pyruvate decarboxylase, which is converting pyruvate to acetaldehyde, and then you are using NADH, 2 molecule of NADH, so that NADH can donate the hydrogen into the acetaldehyde structure, so that you can generate ethyl alcohol. So this is another fate of pyruvate, and the third fate of pyruvate is the aerobic oxidation, which occurs exclusively in all aerobic cell or aerobic tissue like aerobic bacteria like us, etc. Where pyruvate is getting converted to actually acetyl-CoA, and acetyl-CoA gets incorporated into the TCA cycle, that is alternately called the citric acid cycle. So pyruvate is getting converted to actually acetaldehyde. Sorry, pyruvate is getting converted to acetyl-CoA, and acetyl-CoA gets incorporated into the citric acid cycle. So citric acid cycle, why it is called citric acid cycle? Because the first component, the first product of the TCA cycle is the citric acid, and that is why it is called citric acid cycle. The citric acid cycle starts with a condensation reaction between oxaloacetate and the acetyl. So the reaction by which you are converting pyruvate to acetyl-CoA, that is not written here, pyruvate to acetyl-CoA. This particular reaction is actually the intermediate of the glycolysis in the TCA cycle. So the conversion of pyruvate to acetyl-CoA is a 2-carbon unit, and oxaloacetate is a 4-carbon unit. So this 4-carbon unit oxaloacetate is already a part of the TCA cycle, and we are getting the acetyl-CoA. So the acetyl-CoA and oxaloacetate combines together to form the first compound, which is called the citric acid. So that is why it is called the TCA cycle or citric acid cycle, and TCA means tricarboxylic acid cycle. Actually, in citric acid there are 3 carboxyl group present in the structure, so that is called TCA cycle. So after TCA cycle or citric acid cycle, we need to generate, there are several conversion of NAD to NADH and FAD to FADH2, which should be incorporated to the electron transport chain and oxidative phosphorylation, where those NADH or FADH2 will be converted to NAD or FAD actually, and for this process we require oxygen, and ultimately we are producing 6CO2 and 6H2 by this process with the concomitant production of ATP molecule. So in class 9 or class 10 life science book, you will read that under aerobic oxidation 32 to 30 molecule of ATP will be generated. It is actually 36 or 38 has been written, but actual number is 30 to 32 molecule of ATP, only 30 to 32 molecule of ATP. Under complete aerobic oxidation of glucose, only 30 to 32 molecule of ATP, 30 to 32 molecule of ATP will be generated, and this process is carried out by the oxidative phosphorylation pathway. So ultimately, glucose plus 6O2, you are using 6O2 here, so glucose plus 6O2 equal to 6CO2 and 6H2. So this is the overall of glycolysis and the anaerobic fate of aerobic and anaerobic fate of pyruvate. Okay. Hope you understand the introductory part, and in the next video we are going to discuss about the introductory the details of the glycolysis, the first step of the glycolysis, and the technical steps of the glycolysis, which is known as the preparatory phase of glycolysis and the ATP generating phase of glycolysis, right?